Hi viewers, in this class we are going to discuss the precipitation reaction apart from the antigen antibody reactions. Under this topic we will discuss the basic introduction, mechanism of precipitation, pro-zone, peak zone and post-zone, common precipitation tests and applications of the precipitation reaction. Precipitation is an antigen-antibody reaction between a soluble antigen and its specific antibody that results in the formation of insoluble precipitate. Precipitation of antigen-antibody complexes occurs because the divalent antibody molecules cross-link multivalent antigen molecules to form a lattice. When it reaches a certain size, the antigen-antibody complex loses its solubility and precipitates out of the solution. The phenomenon of precipitation is termed as a precipitin reaction. That is, it occurs in the equivalence zone. Precipitation is defined as when an antibody interacts with soluble antigen, it will result in precipitation. And the antibody is known as precipitin. Antibody is known as precipitin. While the precipitation occurs in equivalence zone, where equivalence zone means the concentration of antigen is equal to the concentration of antibody. Meanwhile, it does not carry out in AG excess, that is antigen excess and antibody excess zone. Marak in 1934 proposed lattice hypothesis to explain the mechanism of precipitation. According to this concept, multivalent antigen combines with bivalent antibody in varying proportions. Precipitation results when a layer of lattice is formed consisting of alternating antigen and antibody molecules. This is possible only at zone of equivalence. In the zone of antigen and antibody excess, the lattice does not exist. So here we are observing the precipitation reaction when the soluble antigen combines with its antibody in the presence of electrolytes at a suitable temperature and pH. The antigen-antibody complex forms an insoluble precipitate. Precipitation can take place in liquid media or in gel. Instead of sedimenting, if the precipitate remains suspended as floccules, the reaction is known as the flocculation. See here, this is a precipitation curve. This is the zone of equivalence where the concentration of antigen and antibody are equal. This is the zone of antibody excess. More antibodies than the antigens, and here zone of antigen excess. Antigens are more than the antibodies. When increasing concentration of antigen are added to a series of tubes that contain a constant concentration of antibodies, variable amounts of precipitates have been formed. If the amount of the precipitate is plotted against the amount of antigen is added, a precipitation curve like this show three important zones. That is the equivalence, zone of equivalence, that is the proportions of antigen to antibody is optimal for maximal precipitation. In the zone of antibody excess or antigen excess, the proportions of the reactants do not lead to efficient cross-linking and formation of precipitate. The amount of precipitate formed is dependent on the proportions of the reactant antigens and antibodies. The correct proportions of the reaction result in maximal formation of the precipitate. Excess of antigen and antibody results in soluble complexes. The precipitation reaction can be performed in fluid phases By mixing the aqueous solutions of antigen and antibody in a test tube, a cloudy precipitate ring forms. A cloudy precipitate ring forms at the interface between the solutions of antigen and the antibody, where the ratio of antibody and antigen concentration is optimum. This area in which the ring forms is called as the zone of equivalence. So here this is all the antigen zone and this is all antibodies where the precipitation has been formed. This is the zone of equivalence. Zone of equivalence. And the test is known as the precipitation ring test. Here where we will see the peak zone or zone of equivalence. In the middle, this is the antibody excess zone and this is the antigen excess zone. The precipitation reaction is the basic principle in the assay techniques such as the single immunodiffusion, double immunodiffusion, radial immunodiffusion, immunoelectrophoresis, rocket immunodiffusion and the immunoblots like the western blots. And there are common precipitation tests, namely ring test or interface precipitation test, slide test, VDR slide test for syphilis, tube test. Apart from that, precipitation test in gels, that is immunodiffusion and the immunoelectrophoresis. Apart from the immunodiffusion, single diffusion in one dimension, double diffusion in one dimension, single diffusion in two dimensions and double diffusion in two dimensions. Let us see what are all these tests briefly. This is the precipitation test or ring test. It can be done by taking a small test tube containing antisera and place a layer of antigen above it. Here it is antisera and we have placed the layer of antigen above it. Then the antigen antibody diffusion occurs inside the tube and forms visible precipitate in the shape of the ring at the intermediate zone of both of them. That is nothing but there the equivalence zone. And the best example for this is VDRL test. VDRL test for the syphilis. VDRL means that is the venereal disease research laboratory test is designed to assess whether you have the syphilis, that is a sexually transmitted infection. Syphilis caused by the bacterium Treponema pallidum. The bacterium infects by penetrating into the lining of the mouth or genital area. The test detects the presence of antibodies against the bacteria Treponema pallidum. The VDRL is a blood test for syphilis was developed by the Eponymous lab. The VDRL test is used to screen for syphilis. It has high sensitivity developed by August Paul von Wassermann with the aid of Albert Neisser in 1906. In 1906. The VDRL is non-treponemal serological screening for syphilis that is also used to assess response to therapy, to detect central nervous system involvement and as an aid in the diagnosis of congenital syphilis. The base of the test is that an antibody produced by a patient with syphilis react with an extract of ART, that is the diphosphatidylglycerol glycerol. It therefore detects anticardiolipin antibodies IgM or immunoglobulin A visualized through foaming of the test tube fluid, that is flocculation. The VDRL test doesn't look for the bacteria that cause syphilis. Instead, it checks for the antibodies your body makes in response to antigens produced by cells damaged by the bacteria. Here, slide test. When a drop of antigen and antibody are placed on the slide and mixed by shaking, floccules will appear between antigen and antibody. So the clinical application is VDR test for syphilis. Immunodiffusion. In this method, the specific antigen and antibodies are allowed to combine in a semi-solid or gel medium. Immunodiffusion is performed by using agar or agarose gel. What are the advantages are? The reaction formed from this method is stable and can be preserved for staining. It can be used to detect identity, non-identity and cross-reaction between antigens in a mixture. Single diffusion in one dimension. Here, the antibody is incorporated in a agar gel in a test tube and then antigen is layered over it. The antigen diffuses downwards through the gel, forming a line of precipitation. Precipitate dissolves as the concentration of the antigen increases. The number of bands indicates the number of different antigens in the test tube. Then single diffusion in two dimensions. Single diffusion is in two dimensions is also called as a radial immunodiffusion RID. It has been used for the quantitative estimation of antibodies and antigens in the serum. Double diffusion in one dimension. It is also called as the Oakley-Falthrow procedure. In this method, plain agar is also used. AB antibody is incorporated in a gel above which a column of plain agar. The antigen is layered on top of the agar medium. The antigen and antibody move towards each other through the intervening column of plain agar. Thereby, this will result in the formation of the precipitate when they meet at optimum proportions at the middle of the tube. Double diffusion in two dimensions. This is also known as the Octorloni procedure, known as agar gel immunodiffusion or passive double immunodiffusion. In this method, both antigen and antibody diffuse independently through agar gel in two dimensions, horizontally and vertically. The technique is named for the Orjan Octorloni, the Swedish physician who invented this test in 1948. The procedure is as follows. The test is performed by cutting wells in the agar gel poured on a glass slide or in a petri dish. The antibody is placed in a center well and different antigens are added to the well surrounding the center well. After an incubation period of 12 to 48 hours in a moisture chamber, the lines precipitants are formed at the site of the combination of antigen and antibody. And coming to the immunoelectrophoresis. Immunoelectrophoresis is the migration of molecules due to electrical charge. Positive particles travel to cathode, negative particles travel to anode. Precipitant specificity provides a critical indicator of the identity. When the concentration of fraction of serum such as albumin alpha, beta, gamma has to be determined, this test can be carried out. Take a gel slab, place the serum, conduct electrophoresis. The fraction gets separated according to the charge and size. Make a trough place AB and AG and AB diffuse, antigen and antibody diffuse from visible precipitate. To elaborate, in a rocket electro, the visible PPT precipitate is in the shape of a rocket. The weight of the rocket is directly proportional to the concentration of the antigen. But it should have a negative charge, whereas it counter immune electrophoresis. It can have any charge. It can be carried out in two different ways. So this is all about the immunoelectrophoresis. And coming to the applications of precipitation reactions. It will be carried out as qualitative and quantitative for detection of antigen and antibody. It is very sensitive test for detecting antigens, relatively less sensitive for detection of antibody. Capable of detecting the little quantity of antigen protein as 1 microgram. And it is used for identification of blood and seminal stains and food adulterants. These are the applications of precipitation reactions. Thank you one and all.